clarify to this group what it means to express youthful gene expression. From the time we were created till around the age 25, our gene expression profile inside our cells was very healthy. As a part of normal aging, the beneficial genes of youth start to be suppressed. Unfortunately, the bad ones start to be expressed. So we have a chronic problem of deleterious genes overly expressing themselves while the youthful beneficial genes are being suppressed. Now, the ultimate cure for aging will ultimately be total control over gene expression. We could make all the telomerase we want. We can suppress cancer cell proliferation. We would be in total control of our cells the way we are of our computers. We'd be able to make our cells do what we want them to, which is to live indefinitely. Now, we don't have Dr. Church's technology available yet. His CRISPR-Cas9 is still in a research mode. They're projecting that's 10 years away. We can't wait 10 years. So we have people like Liz Parrish, who has been investigating ways to use viruses and other methodologies to alter our gene expression so it starts to behave like a younger profile. And this is incredibly important because this could be the connection between what technologies exist now, which of course are limited, people are still dying, and 10 years from now when we may gain total control over aging. So what Liz represents is an important stepping stone between today and perhaps 10 to 12 years from now when aging is a controllable, managed situation. So I want to welcome Liz Parrish up to this stage and also applaud her for the courageous work she's doing, because she has been doing self-experimentation and she's been on the front lines of it.
Thank you, Bill. Wow, that's amazing. And I'm sandwiched between two great speakers. Thank you so much. I'm missing my first slide, but that's okay. I'm glad that you guys are excited, and I think you have a lot of reasons to be excited. The future looks bright, and the future of gene modulation looks even more amazing. So right now we're working on health genes, but in the future we might be genetically engineering you for where you want to live, what you want to do for a living, and how you like to spend your free time. In order to do that, BioViva is working hard to create an ecosystem where we can take you, help you understand the process, have patients go through, and then come out with data that is actually meaningful. So how we're going to do that is we're working with human genomics. BioViva next month launches its first DNA sequencing. So you'll be able to get your SNP testing, you'll be able to get your whole genome sequencing, and if you've already gotten it, we're offering genomic counseling. So that's really exciting. Comparative biology, we look at all organisms. So we look at organisms that are used as model organisms in science, and we look at organisms who have longevity beneficial genes. There's actually somebody is talking through this monitor. Sorry, it's distracting me because somebody's voice is actually talking back at me. Then gene therapy, creating a platform for you to take gene therapies and find out what happens through analyzing the biomarkers of aging, and finally longevity counseling, which we already talked about. So this looks like a really crazy map, and it is at first glean. There's about 200 genes that are associated with aging, genes that get turned on over time and expressed differently as we age. And there's about 59 longevity-associated genes. So those are the genes that we're looking at that will be a benefit to humans. Telomerase induction. So this is one of my favorites because I participated in this gene therapy. Telomere lengthening has a lot of benefits. So repairing telomeres restores mitochondrial function. It improves genomic stability, and it reduces senescent cells. Maria Blasco recently published a paper that was pretty exhaustive and showed, like you'd heard before, that telomerase induction not only doesn't cause cancer, but may actually protect against it. Follistatin. Follistatin is another one of my favorite genes. It's a gene therapy that I partook in, and it is a gene that actually increases your muscle mass. So when we increase muscle mass for patients who are older, who might have sarcopenia, frailty, they might suffer from cachexia from taking a cancer therapy, we give them back their life. We give them back their energy, their life, their strength, and their ability to get back to doing the things that they really love to do. This gene therapy has actually been in over 30 patients, and Pfizer actually just launched their version of it, and it's helping kids with muscular dystrophy climb stairs, be active, and get back to a normal life. Sorry. Enter the goddess Clotho. Clotho is a gene therapy that we have a lot of hope for. So patients who had renal failure were found to have low levels of the protein from Clotho. Clotho regenerates muscle, it improves brain function, it suppresses growth pathways that we don't want to have. It upregulates two genes that we're also interested in, which is FGF21 and FGF23. These are actually fibroblast growth factors, and they help regenerate areas of your body. So Clotho is a fantastic therapy that we think will help people with kidney failure initially, and then we'll move it on to whole body regeneration. About 1 in 7 people will have kidney failure in their lifetime. PGC1 alpha. This is a great gene. So when we look at PGC1 alpha, we're looking at mitochondrial function. This is a gene that reduces white fat, increases energy production, and increases brown fat, which is actually beneficial to the body. One way that we can think of PGC alpha is when we used to look at animal studies, we'd look at things like mice, and we'd say, okay, a mice per gram of body weight has 7 times faster metabolic rate than a human. So we used to say that things that burn fast, burn bright, burn fast. Lots of energy, shorter lifespan. But in fact, a bat, who is closely related to a mouse, lives to about 41 years compared to the mouse's 2 years. And what that means is we want to look at what's happening in those cells, and PGC1 alpha is the bat's key to success. So when we're looking at comparative genomics like we just did, we're looking at animal models, we're looking at animals that live longer, that have benefits, that may not suffer from cancer or heart disease or other diseases that we prominently suffer from. And we can actually look at this as a phylogenetic grocery store. So we can look at these type of technologies as something that we can incorporate into our own body over time and with testing. This is a great video by UCLA, and this is what gene therapy delivery looks like. So this is actually AAV2. AAV2 is a delivery method for gene therapy. This shows how it gets into the cell, how it then goes and attaches to the nucleus, injects the DNA, and then the DNA creates an episome. It doesn't integrate into the human chromosome, and then it upregulates the protein. AAV is a great gene therapy. It's in 140 clinical studies. It has a low immunogenicity. It actually can be designed to only target certain tissue types, and that makes it really exciting. But of course, at BioViva, we're all about bigger, better, and more versatile. So this year, we're funding research to create another delivery method for gene therapy that is bigger and better. It holds more genes, and it's as versatile as AAV. So if I put things into perspective, AAV can get in a very small gene. Our new delivery method could get in all 4 genes that I just described to you. So that is a big deal. Delivering more genes at one time is beneficial. It would be more cost-effective to patients. It would be a way that we can actually customize a gene therapy for you. So different people will have genetic weaknesses that we might want to compensate for whilst we upregulate regenerative gene therapies. Biomarkers. So actually, I just did another telomere test this year, and my telomeres are longer. So some of you know that I did the 2 gene therapies, a telomerase inducer and the myostatin inhibitor, and we were actually thrilled to find out in the 3rd year that the telomeres lengthened once again. So when I first started my journey, my telomeres were extremely short. They were about the same as the approximate age of a 65-year-old. After therapy, 1 year later, they were about my average age, about 45 at the time. They've actually lengthened some more, and they're the average age of a 30-year-old. Now, remember, this is just my T lymphocytes, so, but this is a good place to start. These are the cells that they look at, and they're a good model for treating aging. My muscle mass increased with the myostatin inhibitor. We're about to go and get yet another test, and we're really excited to see what's going to happen next with the muscle mass. I actually went from exercising regularly to not, and we still saw increase in muscle mass and decrease in white fat, which was really great because we all want to live our lifestyle. But we learned a lot from my test. We learned that the biomarkers of aging were greatly lacking, and we need to do more to actually envisage what's actually happening with patients over time. So recently, we just put money into hiring a whole bioinformatics team. We're going to be analyzing as many biomarkers that we possibly can to not only find out what's happening in the general population, but find out what's happening with patients that go through gene therapy. So these are the physiological biomarkers of aging that we'll be looking at. And they go from everything from strength to functional tests to B cells and C-reactive proteins, which are a sign of inflammation, and it's a pretty exhaustive list. We're also looking at molecular biomarkers, and this will be of great benefit, especially to looking at what's happening in proteomics after we take a gene therapy. Anatomical and phenotypic markers are really important, and just so that you know, when we think about phenotypical markers, we think, how powerful could that possibly be? But the imaging through a body, things like MRI and skin imaging, are actually really powerful tools to know how well we're doing all the way through. When we look at images of old bodies, we see atrophy and detriment all the way through the body. This will give us the ability to look at the human body in a new way and create an atlas that will show us what is actually happening during aging. And when we actually reverse that aging, we will have great biomarkers to express to the world. So a bioinformatics map is putting together all of the omics, and this is what our team is doing. So we're working with public sources of data. If you look at the UK, they're running the 100,000 Genome Project. People are running data on the microbiome. We are continually running data on what's happening to people who live in certain places in the world with a certain amount of pollution. And we're going to combine those to give the best reading that we possibly can on what's happening in the human body. Longevity counseling will be starting in 2019. So this year, what we've done is we've actually opened up through our company, Integrative Health Systems, to treating patients with gene therapy. So I can tell you that the doors are open, the doctor is ready, and we've already treated patients. So in the next year or 18 months, we'll start to have some data. We're going to wait till we have some substantial data, and we're going to start using that data to help you understand more what's happening with your health over time. And that will work into this longevity counseling that will help you actually map your future and create better outcomes for your health. Thank you so much for having me, and I think that I come on the precipice of another great speaker. Thanks a lot.